the screen
Yeah.
they're on. Thank you. And we have a clicker somewhere. Let's get that. Perfect. And what else do we need? Da-da-bum-bum, ba-dum-bum. Who's up first? Ryan? Yeah, yeah.
I know. That much I know. I have one video in the presentation, but
Have
if
you
it
tested it?
yes, it worked in the PowerPoint.
Which presentation?
The second.
The second. We can check.
Yeah.
Are we presenting in the same order as is at the...
Ryan, insert James, Marco, and Monica.
Yeah, that's
So
Ryan,
that's the
insert
order,
James,
right?
Marco, and Mike. Yes.
Great, thank you.
Love it. What's done? 5 past. Yes, I know, yes. Yes, yes, we're all Wi-Fi. Sorry, yeah.
No, we're up here. Don't worry.
Yeah, okay. Yeah. We'll give it a few more minutes.
Yeah, I left all my friends back.
Here and across the world, potentially. Welcome to the final session of the final day. 3D TV may have been a fad. No one wants to wear 3D glasses to watch their movies, but 3D tissue, that's no fad. That is the next thing. So in light of that, we've got 5 speakers for you. Sorry, I'm sorry. I'm used to not using a microphone. I've got 5 speakers lined up for you today. And they're going to be talking about their investigations in innate immunity in real microgravity with advanced bone marrow organoids. We've got human cerebral organoids looking at neuroscience research in Earth and space conditions. And we're looking at mechanobiology as well with bioreactors. Further, we have some ground analysis on mechanical actuators and electrical sensors in 3D organs on chip models. And last, but by no means least, we have a talk on the ex vivo and in vivo 3D models for space research. So with that, I hand the floor over to Ryan Sakar, who is going to be talking to you. Well, he'll introduce himself. Thank you very much.
All right. Thank you. It's lovely to have a hype man. Yes, I would need that. Yeah, so I'm Ryan Sarkar. I'm from Goethe Uni Frankfurt. I'm going to talk to you about the Immuno3D campaign and the SHAPE experiment today, which is all about bone marrow. So, is this? Nothing is happening. Press really hard. Oh, okay, now I'm like in the middle of my slides. Cool. Okay. Yes. Why is this animated? Anyways, the title of my slide, when the animation is finished, everyone will be back from lunch. It's all good. Yeah, so investigating innate immunity aboard the International Space Station using human bone marrow organoids. And I'm part of a team with Louise Breiteband and our PI, Francesco Pampaloni, I'm standing in here for him today. Yeah. So, as we all know and have heard multiple times over the last few days, astronauts are now planned to go farther and farther and farther from Earth. The thing is, and this I'm sure you have not heard at all in the last 3 days, no one has told you this, but they suffer from bone density loss and immune dysfunction. So the issue is how do we mitigate these issues when really we're still trying to understand them? So given that all your innate immune cells come from your bone marrow, the Immuno3D experiment was devised to basically figure out if these two issues are related. So what we have is we take a high-fidelity model, a physiologically relevant model of the bone marrow, we make an organoid from it, and then the plan is to fly them to space in a high sample size. As in vitro experiments on the ISS, they tend to be either 2D cell cultures or suspension cultures, which aggregate randomly. Here we take a standardized organoid model, and we fly 2,500 of them because a lot of the literature is very contradictory, because you get small sample sizes, every space flight is different, hardware setups are different. So we wanted to try and address that as best as we could with our own experiment. And so that's where this SHAPE experiment comes in, which flew to the ISS in November 2022. So how do you get from bone marrow to organoids? This is simplifying it a lot, but inside the bone marrow, we have the stem cell niche, which has, I'm going to focus on two main cell types. We have mesenchymal stem cells, which can do a lot. They can migrate a bit. They can help heal tissue damage, as we heard in the previous session. And they can also give support to other cells. And so in the stem cell niche, you have mesenchymal stem cells, which are in contact with hematopoietic stem cells. Now, these are very important because they become myeloid progenitor cells, and they differentiate further into the major components of your innate immune system, your body's first line of defense. The thing with hematopoietic cells is that they're difficult to keep in vitro because they like to differentiate. They basically don't want to stay progenitors. But the mesenchymal cells, when they're in contact together, can keep these cells immature. So then we developed this organoid model where the mesenchymal cells and the hematopoietic stem cells, they're all primary stem cells sourced from individuals. We make an organoid model where they self-aggregate, they're in contact with each other, and then as 50% of the bone marrow is bone mineral by weight, we also add these micrometer-sized hydroxyapatite beads to help simulate the inorganic component of the bone in case the mesenchymal cells interact with that, and that gives a higher physiological relevance of the model. So we take these organoids. This is how they look like. The beads are just incorporated really easily. And then the hematopoietic stem cells organize themselves into a niche, which is why we call it an organoid and not just a spheroid, because they're not just located randomly throughout the bone marrow organoid that we made. So how do you culture these in space? On Earth is tricky enough. So we developed this agro system called the Hydra well. It's kind of like a U-well. In it, you can seed the cells. The cells will then aggregate themselves due to gravity. And then the beautiful thing here is that you can fixate, you can image, you can do your entire experimental workflow without ever disturbing your precious space samples. And if you stagger when you seed the cells, you can get organoids that are actually formed in the bottom hydra well, and the one on top, if we seed the cells later, you can get cell suspension, which is what our experimental design was. So we could look at organoids and how they behave in microgravity in space, but also how do these cells aggregate and how does microgravity affect the formation of the organoid in space. So then this was supposed to, oh, what's happened? No, I didn't press anything. My slides just disappeared. Oh, that's a big rewind. I can just skip ahead again. Yeah, so here was supposed to be. We can skip this slide. No worries. If it's causing issues, we can skip that one. And I will just adapt. All right, everyone cross your fingers that it doesn't crash. Yeah, I'm not, he's controlling it for me, so. Yeah, I just start the presentation after that slide. Or delete it or something, I'm not sure. Sorry about that, folks. but I can continue talking. So anyways, the organoids are made in 1G. So they're in this hydrowell like little sandwich. They're self-contained. And that way, when they go in the hardware, our hardware was not designed specifically for us. We're using older hardware that's been used in experiments before, already flight tested, which made the preparation of this entire experiment, it was ready to fly in like 18 months from conception, but then COVID came and we ended up flying 2.5 years later. But yeah, basically, this gets perfused by the hardware via pumps, and it's all done remotely. And I'm sorry. But anyways, this flew to the ISS in 2022. And I would love to show you pictures of what the cells look like when we got them back. I believe now they're trying to. Hey, we could, worst case, I get my laptop or something, so. Okay. Should I ask them?
If I can delete the slide, by all means.
Yeah, that's what I'm saying. Just delete the slide. That's what I'm saying. I don't really need it to go. Oh, yeah, that's fine. Okay. I will go back to my corner. You would have to ask the session, I guess, Chair.
Surely it won't take long.
Yeah, I would fine. Okay, we'll give him a minute, yeah. Do you have any good space-related jokes I could take? Yeah, for sure. Yeah, let's do questions.
Why did you choose that material in the first place? How did you choose it?
Hydroxyapatite or the agarose? Okay, so hydroxyapatite itself is just like a slightly different form of bone mineral. So hydroxyapatite, yeah, it's like the closest thing that we could buy in like a. It was important to us that it's like in a very small like bead form. You can't really buy bone mineral in a bead form to add to the. No. Like just looking at the chemical composition, we aimed specifically for that one. Yeah, I mean, the agarose we picked because you can change the concentration to choose stiffness. And then it's important to keep the organoids separate from each other because they're mesenchymal cells. They like to stick to each other. And then if you get, in each little hydrowell, it's like 5 by 3 centimeters or something. If you have 70 organoids in here and then they all aggregate to make like one massive conglomeration, you can't put that in the microscope. Everything will die, break apart. It's completely unanalyzable. And it's not a good model at that point. It's much larger than, yeah, so the agarose was important just to be perfusable for medium, for fixatives, but also that the cells themselves won't stick to it. It's also like really cheap and easy to work with, which is like an added bonus as opposed to some, we work with hydrogels too. We could have used some fancy hydrogel if we wanted, but if agarose does the job. And that's why it's also like the hydrogel itself is a good system because you can put other types of cell cultures in there. We also do experiments with gastruloids. There's people using like liver organoids that want to try adapt them for that. And we were thinking like we could probably get gel droplets to even work in there if we wanted, but I'm the only one in my lab working on microgravity right now, so no one really needs them at the moment. No. We have that in our lab. We have bioprinters and such, but for us it was important that because hardware and adapting an experimental design for space has so many challenges that we wanted like the simplest cell culture system possible. So we picked something that organizes itself as opposed to we have to do some work. Yeah, skip that one. I don't want to see it. Yes, okay. So This is what we had to use no-tox-histo. I don't know if you guys know what this is, but it's a non-toxic fixative that's good for the ISS. We did lots of fixative testing. So here you can see, nuclear dye staining. You can see a vimentin antibody staining, and then you can see F-actin, which this looks like it didn't work, and that would be correct. No-tox-histo dissolves the actin cytoskeleton. But stored the samples for 60 days at 4 degrees. The no-tox-histo itself, we slightly modified because if you just use no-tox-histo in the hardware, it will mix with the medium and a 50-50 concentration, and then it makes this precipitate, and you can never find your samples again, and it ruins everything. So just a supplementation of a little bit of hydrochloric acid. No-tox-histo is already very acidic. Then got rid of the precipitation, and then we could look at it. But this isn't PFA, so if you leave your samples there for a long time, 120 days, you can see they start to degrade. Things get a bit worse. I guess now you guys have to scroll down for me. So next slide, please. And this is how it looks like. And then here's what an organoid looks like when it's actually made on Earth, fixed with PFA. That was all cultured in the hydrowell, fixed in the hydrowell, imaged in the hydrowell. And then this is what our hydrowell sandwich looks like before we go in the hardware. So there are lots of unforeseen complications in every space experiment, and I think this very clearly illustrates one that we had to face. One of hundreds of issues that we had, but basically if my system is based all around gravity pulling my cells together, then the rocket, it should be loaded on its side. But we weren't able to choose a rocket in the end. So when we flew Northrop Grumman and Taurus, they're like, is it okay if we put the hardware on its side for 72 hours? To which I said, well, I don't have a choice, right? So, and hydrowells weren't designed to work like this, so I had no idea if my cells would even still be in the hardware, or not in the hardware, but in the hydrowell after. But luckily they were, which you would see on the next slide. So this is what we got back from the space station. Compared to what I showed you earlier, it's a lot looser aggregation. The coloring you see is just, we graded everything based on which Z level it is, just to see how the cells themselves are distributed, whether they actually aggregated themselves or whether they just coated the bottom of the well. So we could go to the next slide. And then, so due to the suboptimal storage conditions, the fixation didn't really work as well as we hoped, but we still got the nuclei. So then we were wondering, what can we do with these nuclei images? And so this is what we did. Given that we can go to the next slide. So given that the mesenchymal cells are much larger than the hematopoietic, so mesenchymal cells tend to be around this big, and then a nice hematopoietic one is there. We thought if we can segment all the nuclei and then make thresholds to divide them based on their size, we can try and figure out what cell type is which and see how they're distributed. So and this is how it looks like on the ground, where you can more clearly see hematopoietic cells are a lot smaller, and then mesenchymal cells tend to be a lot bigger. So if we go to the next slide, then we quantified how far the two cell types are based on their thresholds from the center of the organoid to see if a niche formed. So if you get a tight distribution here of the hematopoietic cells, that corresponds with the hematopoietic niche in the organoid. Whereas in microgravity, which is denoted by my little astronaut, it's just a normal distribution all throughout the organoid, and therefore no niche formed. And of course, the rocket being loaded on its side could be a confounding factor. But that's something, we couldn't really control for. So for now, we're okay, in microgravity, we didn't see the niche form. If we go to the next slide. So half our units were fixed with no-tox-histo for imaging, and half were fixed with RNA later, because we want to do RNA sequencing. But we already expected, like I told you, that we had suboptimal storage condition for our fixatives. So we went with what's called MACE sequencing, which was recommended by a colleague of mine, Dario, which stands for Massive Analysis of cDNA ends. And it's a very sensitive technique, which is good for also degraded samples, because degradation usually starts from the 5' end, not the 3' end. So this is what we did, and we can go next. So in our MACE sequencing, this is the 9 units that we have in total. Up here is the ISS microgravity unit. Then we have the 2 centrifuge controls here. And then here we have our cluster of 1G ground controls. So yeah, there's a cluster of definitely the ISS-1G units, and then there's the microgravity, and it's very distinct. If we go to the next slide, then it starts getting into heat maps. And so here there's like an overall trend of just underexpression in the ISS samples. And on the right, I just have, these are the 50 most differentially expressed genes between all the units. Things in red are for innate immunity. Things in blue are for the structure of the organoids. So like ECM proteins are ECM genes, actin genes, and then things in green are in the middle. And if we move to the next slide, yeah, so this is my favorite slide. So this is a heat map of the 50 highest differentially expressed genes of only the ISS units. And so in red are the innate immunity genes. So I just want to highlight 3 important ones to me. So SOX4 is very important in innate immunity. It's specifically linked with suppression of the innate immune system and then subsequent viral pathogenesis, which is important given that astronauts, we know that they suffer from like latent viral reactivation and they're much more susceptible to these infections. And so this was strongly overexpressed in the microgravity compared to the 1G centrifuge controls. Another gene was ZBTB7A, which again, it's linked to suppression of myeloid differentiation. So these hematopoietic cells cannot go down the innate immune differentiation path when this gene is overactive. And then there is also TRIM56 and SMAD2, which were underexpressed, and these are linked to like the TGF-beta modulated regulation of innate immunity, and so their activity is important for the innate immune response to occur, and then in microgravity, they were severely underexpressed. So if we go to the next slide, now it's just my summary, which is that with the Hydra wells, we managed to fly 2,500 bone marrow organoids to the ISS for 5 days exposure in real microgravity. We wanted like the high sample size, but given our fixatives, we weren't able to take each individual organoid like we originally planned. The Hydras themselves worked perfectly fine. In microgravity, we threw them on their side just as a surprise, and they performed admirably. They've got validated as a system that we could use for other 3D cell cultures. We didn't see the hematopoietic niche in microgravity, but what we did see from the MACE sequencing is that we had genes involved in innate immunity, a lot of genes involved in innate immunity that were differentially expressed in microgravity compared to the centrifuge controls. So if we go to the next one, just want to acknowledge a lot of people at the DLR for helping us. It is basically a small army behind every space experiment. I want to thank my lab, the people in America that helped us get ourselves onto this rocket, because that was a journey in and of itself. And then with that, I guess I'm done.
Ladies and gentlemen, this session will be a panel discussion at the end, so we've already had some questions, but there won't be any questions, and that will help also with time. Insa, you're up next with cerebral organoids.
Thank you. Yeah, we switch organ, and maybe we also switch a bit the complexity. Okay, I have to practice with this as well. Maybe, yeah, can you just start the presentation? Here we go. Wonderful. Yes, so I will talk about a rather young project, which is the Micro-Heavy Brain Organoids project. We at GSI are interested in the brain and the adverse effects that radiation has on the astronauts. And yeah, I put this only up to say that, of course, yes, we are interested in radiation at the GSI, but the space doesn't come only with radiation. So we teamed up with the DLR to also include altered gravity. And I also put this up because, of course, when we talk about acute and late CNS risks for astronauts, that's neither new nor is it unique to the astronauts. So I wanted to make the point that we have thousands and thousands of guinea pigs, more or less, in those patients with neuropsychological disorders. And I point out that we can not only cater to them and help them, but that we can also learn from them. And I think that's even more valuable, because if you look at patients with neuropsychological disorders, like patients with depression, for instance, there are a few brain regions in the MRI that show really severe changes, and that's the prefrontal cortex, it's the hippocampus, and it's the amygdala. And I want to talk today about the prefrontal cortex and the hippocampus a little bit, because if you see those MRIs, what happens in those patients is that the prefrontal cortex and the hippocampus shrink in volume. And the activity of these brain regions is reduced, which leads to a reduced connectivity as well within these brain regions. And the reason for this is more or less that we lose the complexity of dendrites and dendritic spines. And of course, nobody of you wants to donate a brain tissue sample, so we need to rely on organoids. We do this. That's the problem that we have. We cannot start with primary stem cells. We have to start from scratch from pluripotent stem cells, embryonic and induced pluripotent stem cells. And we differentiate them along the line of the brain development, and we reach a point that you may think is very premature and maybe not useful. However, nature does not reinvent the wheel. So also in the adults, some mechanisms of regeneration, neurogenesis, and the likes are relying on the same mechanisms or the same substances. And depending on how long we culture them, we can go a little bit further down this line. These organoids are 4 to 8 millimeters in diameter, so they're fairly big. And they have a lot of advantages. One is we can start out from human and patient-specific cells. This brain tissue self-assembles, so this is really cells reacting to the neighboring cells, the inductive measures of the neighboring cells. And that's, I think, a unique selling point to these. As such, they have an identical genetic background, and they have an identical developmental stage. Because also, that is sometimes a problem between co-culture systems. You use a very immature system and co-culture it with the mature cells, biopsies from cancer patients or whatever else. So that is not only a different genetic background, but also a totally different surrounding. And what we can do, and this is nice, we can generate brain region-specific organoids. We can do this in high scalability. So yes, you heard about the 2,500 organoids. We're not that good, but still a few hundred, so 200 to 300 for a campaign is not a problem. And that means instead of having, let's say, 300 mice in a cage, you have 300 organoids in 396 well plates, more or less. And I want to lead you through how we go about this differentiation and the protocols. So to generate brain, of course, we need first a neural tube. And to generate, for example, these hippocampal organoids, which are most interesting for us, we need to get to the rostral side of that neural tube. And thus, we have to limit the Wnt signaling, which means we have to inhibit it. Then we need the dorsal part and not the ventral. So the dorsalization will be via BMP and now Wnt activation. And then from this, you basically go through the development of the roof plate, and then you go to the development of the cortical ham here in blue, and the choroid plexus in red. If you end up there, then you are too far. And from this cortical ham, of course, we can get then the hippocampal entities, and we can also then get the various regions of the hippocampal structure. And this is how it looks like. So we have to do a directed differentiation. So we use a few chemicals to push them in the right way. And then you see we go from the progenitors, from the prefrontal, from the hippocampal progenitors to the neurons, to the dentate gyrus cells. We have CA1 region cells and CA3 region cells. So we are pretty sure that we can generate these kinds of organoids. And these organoids may be cerebral organoids that represent the entire brain, or the region-specific organoids like the hippocampal and the prefrontal cortex organoids. We use in, of course, our bread and butter system, which is simulated microgravity with clinostats, rotating clinostats, and then the space radiation we either mimic with the iron ions at GSI, or we will next year use also the GCR simulator to be even more close to the real situation in space. However, testing to see what our organoids can do, we already flew them on the sounding rocket campaign Morpheus 14, and we also subjected them to some parabolas during a parabolic flight campaign at now in September. And that ends then in the characterization via immunofluorescent staining and all the microscopy techniques that you can think of. We, of course, use PCR and especially the omics, the transcriptomics and the proteomics. And yes, this requires a little bit of work, because if you have an aggregate of this size, you have to build everything from scratch. And here we have a two-chamber system where we have a fixative in the one chamber and our organoids with medium in another one. They are separated by the membrane. And if you can show the video now, you can maybe see here, if the beamer would do it. Yeah, here you can see that now the membrane gets torn out of this chamber, and the two chambers will simply fuse. And then that's how that looks afterwards. In a test, we had 20 organoids in hypergravity and 20 organoids in microgravity compared to the controls. We did bulk sequencing, and we just last week got those results, those raw data back, so I cannot show you any data of that story yet. And as of today, I know also that the proteomics analysis has worked, and also there we can see what we can get. Of course, this is the prerequisite for the identification of molecular mechanisms that are underlying this whole alterations in the neuronal network. And of course, that's not the end of the story, because also the analysis have to be catered or adjusted to these organoids. Because what we're interested in is, of course, the entire picture within the organoid, which is huge. We are also interested in the structural changes, and those go down to a nanometer scale in the dendritic spines. And for this, we use light sheet microscopy as an overview of how the neuronal network is built up in these organoids. And here you can see a staining of SMI microfilaments, where you can see also the direction here in the different colors. And what we do, or what we are interested in, of course, is also the very smallest structures. These are the synapses, and here we can stain the pre- and the post-synaptic markers. And so you have, within this very busy picture, you have the neuronal network stained in red with MAP2. And but what actually interests us are these little tiny dots that you can see here without the neuronal network. And here, actually, you see one example of a synapse. And then you can also see how far are pre- and post-synapses away. And that, of course, also directs the function. So we can see whether these are altered after radiation and simulated microgravity. And we can not only measure, but also quantify this, of course. So to come to an end, I think that the brain, and especially the region-specific organoids, are a very good tool to elucidate these effects of gravity and the space radiation. And the beauty is that we can identify the molecular mechanisms that underlie these changes. And we are already in the process of identifying biomarkers, and especially biomedical countermeasures. We have collaborations with the University of Applied Sciences in Cologne, chemists that do neurotrophic, or that generate neurotrophic samples, and we test them. And of course, we want to transfer this to terrestrial applications and to see whether maybe also hypergravity is a biomedical countermeasure. And with this, I thank all the teams. This is the team on my side. This is the team on the DLR side, and the funding partners.
Thank you very much. From brains to bioreactors, we've got James.
Thanks for a slightly subdued introduction this time, Ian. So thank you for coming to this session at the end of the day, and hello to everyone joining online. My research is on mechanobiology. I think many people in this room will recognize, particularly for the talks we've heard over the last 2 and 3 days, altered biomechanics in astronauts is a primary driver of astronaut disease and debilitation. A lot of my research is more interested in what the cellular effects of mechanobiology are, so looking down at the cellular and molecular mechanisms that go wrong in a lot of disease conditions and with astronauts in microgravity, and also ways that we can develop mechanobiology-derived countermeasures and therapeutics for the populations of Earth, and to enable astronaut exploration of the solar system. So obviously most of the work that I do is on the ground. For the last I guess 30 years or so, many people have been working on bioreactors for a variety of tissue types and cell types. And so huge number of companies have developed proprietary systems, which are generally centered on a linear motor and actuator, which goes back and forth in a very controlled way with a really nice graphical user interface so you can control your loading profile. And the bit we're really interested in is the bit where you grow your cells. So this is the bit where you have the cells interfacing with the engineering system. So the engineering system is taking and applying the loading, and the cell is obviously responding to that loading. And the core things that people have wanted to focus on in terms of developing new bioreactors, both commercially and as academics, is that, of course, they should be physiologically relevant. And if you're a company, you want it to be as adaptable and versatile as possible so you can reach a larger consumer base. So that's the situation on Earth. If we start to think about what the current availability on platforms like the ISS is, it is necessarily a lot more limited. So traditionally, we have Cubic, the workhorse of ESA for the last 2 decades, which is an amazing platform, but potentially coming towards the end of its useful service life. A lot of it is starting to become a little bit outdated and towards obsolescence. We have other companies that are now working on platforms such as Ice Cubes, a partnership with ESA, to develop very adaptable platforms that can be used for a variety of different conditions. But for anybody who was in the Bioculture FDT talk first thing this morning, you realize that ESA's ambitions are really into more integration. So what's the next platform technology for bioscience, cell culture, mechanobiology, 3D tissue engineering that we're going to be using up until 2040 and beyond? And the constraints for this, a lot of them are in terms of space efficiency. Excuse the pun. We want something that's like a multi-tool, and it has to be resource efficient, so we're not using colossal amounts of consumables to run a single experiment, and space efficient, so minimal footprint, and the actual infrastructure has to be able to do a whole load of versatile different types of work. It was a similar situation that I found when I first started my lab in Liverpool about 10 years ago, coming up to 10 years ago. We had a bioreactor. We had this ESA-TC3 system. It's built in Europe. It's a Spanish company. It's really useful. But what we found is that it didn't really have the right cell culture chamber. So the actual biological interface to put our cells into wasn't really suitable for what we wanted. So I had a really good PhD student at the time, Adam, who developed this 3D printing strategy where we can basically put these back into the drawer, the box that it came in, and 3D print our own entirely bespoke cell culture chamber that could culture the cells, in his case, tissue-engineered tendon, in exactly the way we wanted, with increased N numbers and increased experimental power. The benefit of doing everything with 3D printing is that you don't have to fiddle around with screwdrivers and metal parts. It's literally Lego, so everything clicks together, and you can pour all of your cells in a hydrogel into this little mold, and it contracts around these constraint pins here and forms a perfect little tendon. So it's a very adaptable system. And just taking this tensile tendon example as an example, we've actually been able to take this same platform technology of 3D printing and adapt it very slightly to a whole range of different use cases and applications, all the way through looking at tendon in more detail, through looking at muscles and how muscle stretch is important, and also pelvic reconstruction mesh fibrosis, which is a big topic, particularly in the UK, a result of a whole legal cases, why this, under some tensile conditions when implanted into the body, causes extremely painful fibrosis. So we found that this 3D printing strategy, we can take what is basically a platform technology, adapt it with 3D printing, and suddenly it's a whole more useful. And we've also found that all you need to do is actually change a few of the connectors. You can print it with those extra connectors. And this now adapts and connects not only to the ESA-TC3, but also Bose, Instron, and Mechanoculture. So 4 of the most commonly used platforms, one single 3D printer platform that anybody around the world can just download and print. In actual fact, it's even simpler than that, because we find that we don't even need to redesign these chambers. If we've got a nice little attachment bit, all we actually need to do is print an appropriate anchoring mechanism, and we can adjust the morphology of these pins to suit different loading conditions for any tissue type that we want, muscle or tendon. And Adam is responsible for developing a lot of this for different applications. And then the standard grips if you were looking at clamping some sort of material. So a very useful platform technology, which I'm going to come back to at the end of the talk. But the thing that I'm most interested in, and have been working on since my postdoc days, is actually dynamic fluid pressure. It's very commonly called hydrostatic pressure, dynamic hydrostatic pressure. I was interested in this originally because it's considered to be the most important stimulus for bone. So the osteocytes in your bone, as you load them, they sense changes in fluid pressure in the canalicular spaces, and that's really what drives a lot of bone remodeling. About 20% of our bone is remodeled every year. And this is really due to body weight and exercise acting on the skeleton. Of course, when we're in microgravity on the ISS, we don't have those. And there was a whole load of talks, particularly on day one, about physiology and resistance exercise, different types of resistance exercise being performed by astronauts. So back when I was a postdoc, I developed this bioreactor system which uses pneumatic force. So all you do is dynamically compress the gas phase, the headspace of your cell culture environment, and you can do whatever stimulation regime you want to simulate different types of exercise, different types of loading force. And this is, of course, also interesting because there's a lot of other fluid changes in astronauts in microgravity that impact tissues such as the eye. So very important skeletal force in terms of skeletal remodeling and bone health, and fluid pressure changes are generally interesting to study in microgravity in terms of their role in disease. We found that dynamic pressure in a variety of different model systems, so embryonic femur growth and mineralization, and stem cell seeded, these are mesenchymal stem cell seeded hydrogels, is a really great stimulus for driving a lot of physiological responses and physiological bone growth. And we've also found that many cells, particularly stem cells, are extremely sensitive to hydrostatic pressure delivered through pneumatics. This is cells growing after one single low loading event of just 1 hour at a physiological walking pace. And if you hit them with that, they start moving twice as fast and start doing things. And there's a variety of interesting stuff. We also find that after that single stimulation event, so 1 hour with the physiological level of hydrostatic stimulation, we get whole changes in the transcriptome, and a lot of these changes seem to be associated with stem cell niche maintenance. So a lot of the normal physiology of stem cells in the body is regulated by this constitutive mechanical force, which we can simulate with bioreactors of this type. So joining the two strands of what I've been talking to you today in this short talk about, we can actually now 3D print pressurized vessels. So it can combine both strategies. The usefulness and the utility of additive manufacturing, computer design and 3D printing, and the physiological relevance of hydrostatic pressure as a stimulus for cells. So this is a DIY bioreactor that I had a master's project student work on. So we can actually simulate cartilage organoids up to 3 times atmospheric pressure, and potentially up to 7 or 8 times atmospheric pressure just in 3D printed chambers, which is quite interesting. A lot of people thought they would explode, so we had to hide behind the desk when we put it on, but they're fine. We're also looking at how we can do it in more 3D environments, particularly for applications in bioprocessing, cultivated meat, where we have large volumes of fluid that we want to control and apply mechanical forces to. And because pneumatics and hydrostatic pressure is a uniform universal force which acts very similar to water depth, you can basically simulate a lot of those same forces. And regardless of the size of your vessel, you can stimulate biomechanics. You don't have to have point loading, like when you're doing tissue engineering tendon, for example. Regardless of the size of your vessel, you can apply the same levels of universal mechanical force up and down in cyclic, sinusoidal, or whatever waveform you want to stimulate your cells of interest. And then more recently, I've been working with some colleagues on other cell types. So I'm particularly now interested in ophthalmology, and which is extremely relevant. There were some talks during this workshop about eye conditions that are prevalent in crew. Obviously, fluid pressure in the eye is very disturbed and distorted during extended spaceflight, and so we've now found that we can actually print pressurizable vessels, look at some coating regimes to really preserve the life cycle of these. But this is for looking at glaucoma. So these are trabecular meshwork cells, which are important in draining fluid pressure through the front of the eye. And we now have bioreactors that can simulate a lot of these static dynamic flow conditions in eye diseases that are really relevant for the population of the Earth, but will also be interesting to study and develop countermeasures for crew, particularly in longer duration space missions. So this is really all I wanted to talk about today in terms of developing some platform technologies, because I think that's most useful as we're thinking about going forward as a scientific community. What are the platform technologies we need to develop for the next generation of space research post-ISS and post-Cubic? And the other thing that I didn't quite get a chance to talk to you about was mechanical simulation using magnetic nanoparticles, which is also mechanotherapy, which we're taking forward into clinical trials. There's a link to all the publications for anybody who wants to follow up on what the actual biological characterization data we have from all of these platforms is. Here's an example of some of them. So obviously, whilst we're talking about platform technologies, these have all been fully biologically characterized with very extensive data sets. I'd like to thank some key members of my group, particularly Adam, as I mentioned, who's been instrumental in developing 3D printing through a lot of these technologies for Earth and space, and all the people who do the fancy engineering that I'm incapable of. So thank you very much for your attention, and I think the questions at the end.
All right, who's hungry? Let's have some organs on a chip.
Thank you. Hi, and thanks for the invitation to the organizers. I will go on with what James has started, mechanical actuation. So for those of you who are not familiar with organs on chip, well, organs on chip are a mix of biology and technology, and they allow to create biological models which are relevant using some ingredients like human cells, 3D microarchitectures, 3D cell culture, or multiple co-culture systems, and the application of physiological stimulations, biochemical, mechanical, physical in general. So they are not just faster, better, and cheaper, but they have multiple opportunities and give many opportunities, and they have many applications, starting from the drug development process to precision medicine, or the ultimate goal of reduction of the animal testing. And of course, there have been many flights for them to the space, basically provided by NASA so far. So organs chip in our group, by background, I am an engineer, so I'm not a biologist. So I like to play with the technology to create biological models. And we mix together human cells. We provide cells with a three-dimensional environment. We're using mostly hydrogels. And we apply mechanical forces. We do that with a technology which is called UBIT, and basically we provide a mechanical stimulation thanks to a pneumatical actuation. The blue volume is basically gel plus cells. You have to imagine that here. And based on the dimension of hanging pillars, we can translate this compression into either a strain or a confined compression, which is perceived by cells. And the gap underneath the pressure, the pillars drives the intensity of the mechanical actuation. So this technology has been now commercialized by Biomimics, so I have to disclose that I'm a co-founder of Biomimics. So in our group, we are basically agnostic. We work on technology, we like to collaborate with the biologists, and that's why we work on different models. Today, I wanted to go very quickly in two models. One is the heart, which is our first model implemented. So we started with the mechanical stimulation, started with a mix of cardiomyocytes that are from iPS cells and fibroblasts in a mix of 3:1. And we applied mechanical stimulation with a 1 Hertz frequency for 1 week to train cells. And then we analyze them. What happens is that after 1 week of training, the static cells beat, but not in a synchronous way, while the dynamic train cells beat as a syncytium. This is training favors the interconnection of cells between them, and of course also increases the functionality of cells. So we can actually put 2 electrodes in the 2 sides of the tissue, and we can measure exactly the same signal, from which we can actually identify what are the relevant parameters from the heart model. And we can extract those parameters from the electrophysiological curves and measure them. We also measure the maturation level based on ion channels from the PCR to understand whether the mechanical stimulation was actually getting the tissues more mature. So once the model was ready, we actually applied that to test some drugs. So we selected 12 drugs from the SEPA initiative. So SEPA initiative is an initiative to find an alternative method to evaluate cardiotoxicity in a preclinical setting. And those drugs by SEPA are classified based on the potential risks of arrhythmia. So we started with the calibration of the T system with some compounds which are known to affect only specific ionic channels, potassium, calcium, and sodium. And we could actually measure the field potential duration, the amplitude of the signal, and that was a calibration, the calibration that we ran. And we applied the system on 12 drugs based on the SEPA chart, and we could actually detect basically all the potential risks of the drugs that we investigated, classifying them in the proper ranks. So the second application, which is pretty new, it's going to be published hopefully in a couple of weeks, is on gut. So we apply the same technology, basically. Why the gut? The gut is complex, is a complex environment made of different cell types. The main components are the epithelium and the endothelium, which basically are parallel in a villi crypt structure, three-dimensional. There is a mechanical action in the gut, which is very important for our digestion, which makes the gut a very highly dynamic environment. And also there is an additional component which is not present in all the tissues, but mainly in the tissues which form a barrier, which is the interaction with the microbiota, so the bacteria, which is relevant for an application that I will show you in a few slides. So in this case, we applied the system in a different way. So we actually injected cells not in the central part of the chip, but rather in the two side channels to seed the cells in a 2D environment to form two lumens. So one for the epithelium and the other one for the endothelium with a gel, in this case fibrin gel, to emulate the stromal part. And we selected CACO2 cells and a mixture of CACO2 cells and HT29 MTX. HT29 are a cell type which emulates some of the goblet cells, which they produce mucus, which is also important for the bacteria population. So the mechanical stimulation here was actually applied to emulate the peristalsis, so the frequency was a bit less. Here you can see the actuation in a real-time manner, and with the result of a stretching of the barrier. And the training as well did get the tissue more mature. So we have the epithelium lumen on one side, which form like a closed lumen, and the same thing for the endothelium. Importantly, there is a polarization of cells in the epithelium, so the DAPI is on the bottom of the channel, while the tight junction formed are on the top. And the maturation was proven initially with the injection of fluorescent dextran in the two lumen, and the two epithelium and endothelium were formed barriers which were tight for like after 30 minutes there was no diffusion. And also this is proven by the measure of the transepithelium endothelium electrical resistance, which is a measure of the tightness of the barriers. So the mechanical actuation actually, compared to the static condition, formed three-dimensional structures. This was like given only, achieved only in mechanical actuated chips. And we had an upregulation of the proliferation, maturation, and also mucus production in the dynamic conditions. So the clinical question was like to understand whether this system can actually emulate the real patient in undergoing immune checkpoint inhibitor therapy. So we had melanoma patients undergoing immuno checkpoint inhibitor therapies, and we took the fecal samples from the patients, which were classified in either responder or non-responder. And it's known that the microbiota plays an effect on this therapy. So what we got is that non-responder patient, actually, the fecal samples were actually decreasing the tight junction and the cell-cell adhesion markers in the epithelium, as opposite to the respondent patient. And the same things, actually, similar things were observed in the endothelium lumen, where we observe an inflammation, increase in inflammation of the endothelium, providing fecal samples from the non-responder patients. We actually did the same using not the bacteria, but the supernatants. So this to understand whether it was necessary the presence of bacteria or just the supernatants of the samples collected by patients, and the answer was actually the supernatant was enough, and we got the same results. So the non-responder supernatant samples were actually enough to decrease the functionality of the barrier in the epithelium and increase the endothelium inflammation. Just the last slide is on, we asked ourselves whether we could actually reverse this, because there are clinical studies in which the fecal samples of responding patients are used to reverse or to increase the efficacy of the immune checkpoint inhibitor therapy. So what we did was actually to supply a mix of non-responder supernatant together with the responder supernatant, and what we got is a reverse of the effect. So this confirmed that there is an effect of the microbiota a possible effect in the immune therapy, and the same thing happens in the endothelium. So this was a mistake. So this means that we can actually use this kind of devices also to emulate or to go towards precision medicine applications. So just to conclude, we applied mechanical stimulation, and I just showed a couple of examples in which we demonstrated that mechanical stimulation can actually improve the maturation of models. We can actually tune the fidelity of the response of the models to specific tests. As a general guide, the model design, which is a very strict binding of technology and biology, needs to be guided by the specific context of use, which, of course, dictates all the design of process and the selection of specific ingredients. Just to thank my group, especially Mattia, that did all the gut work, and Roberta, who did the heart work, and the financing agencies. And thank you for the attention.
Thank you very much. Thanks very much. Up next, we have Monica. Monica Monici is talking to us today about ex vivo and in vivo 3D models for space research.
So, good afternoon, all of you. So, what I present today is two models we developed in our laboratory. We used these models in experiments of wound healing and regeneration, but I like to present you these models because they could have application also in studies different from tissue regeneration. So we know that the models have a crucial role in research, and in space research in particular, we really are a tremendous needing for reliable models to study the effects induced by spaceflight on the human body and to evaluate the effectiveness of preventative or preventative measures or countermeasures or therapeutic tools. So, we all the scientific community is doing a great effort to move from 2D models to 3D models because they are more correctly representing physiological conditions. So the first model I want to present you is a tissue culture. These are tissue cultures of skin and blood vessels. These models are based on skin and vein vessel biopsies collected from patients undergoing plastic or cardiovascular surgery. So we used these models, we developed these models for an experiment we performed 1 year ago on the International Space Station to study the healing of wounds in microgravity conditions. So after the excision from the body, as you can see, the tissues are stitched to a frame specifically developed to apply a stretching to these tissues. I'm sure you know that after excision from the body, the tissues change their dimension, decrease their dimension, and we saw that to stretch them and report them to the physiological dimension strongly improves the viability of the tissues. So these are phases of the preparation of the tissues, of the culture tissue, and as you can see then for our application we performed a linear wound on the skin, and also we sutured the wound, and so always maintaining the skin stretched. Then, so in the case of the vein, we performed an end-to-end anastomosis, but the important is the culture condition. For the maintenance of the tissues, we culture the tissues at 4 degrees in RPMI modified medium, while for the experiments we maintain the tissues at 32 degrees with MEM modified medium. We modified these culture media adding serelaxin, which is a derivative of relaxin, and metal nonoates, because the serelaxin improves the viability of the tissues, and metal nonoates improve the function of the endothelium. This is the flight hardware we used for our experiment on the International Space Station, but in the labs, you can take these models in Petri dish, simply filled with the culture media. We had very good results from this experiment. We saw several alterations of the wound healing process and also many alterations in the regeneration of the extracellular matrix at the wound site. But what is important is that this model, these tissues cultured in this way survive more than 1 month. So can be, I think, applied for, in several other fields, for example, to evaluate the effect of drugs, allergens, countermeasures of various kinds. And the other model I want to present you is the leech. So also in this case, we use the leech to study wound healing and regeneration. It is a valuable model for studying wound healing because the sequence of events that occurs during this process is very similar to that of the vertebrates. So the leech, from this point of view, is an example of evolutionary conservation of physiological functions that are crucial for the survival of the organisms, such as the healing process. So, moreover, due to the small size of the leech and also the easy maintenance, because they need only water, after feeding, they are able to stay 6 months without feeding any else. So it's very easy to culture them. Moreover, they are invertebrates, so we do not need ethical committee approvals. That sometimes takes a lot of time. Anyway, for our procedures on the animals, we respect all the ethical rules to avoid or minimize animal suffering, and all the invasive procedures are performed under anesthesia. So, in this, we use the leech to study very different kinds of wounds. Here I present a model of wound with loss of substance. As you can see, the wound is performed with a surgical biopsy punch. The dimensions are 3 millimeters of diameter and a depth of 2 millimeters. That, compared to the body of the leech, is a big wound. And then we proceeded with an in situ 3D bioprinting to put a graft in the wound. We used a bioink composed of fibrinogen, nanocellulose, and alginate, and we mixed to this hydrogel human dermal fibroblasts. Another favorable characteristic of this model is that the leech has a very rudimental immune system, so allow us to work with human cells without problems of reject of the graft. Then you can use the model to study the effect of substances that can close the wound, protect it from infections, or favor the healing. Or you can use also the same model to to test the effectiveness of devices that are expected to improve the healing. Or, so in this case, as you can see, the leech is in the chamber of a rotating wall vessel. So in this case, we printed the graft and then put the animal in microgravity to see the difference in the healing on ground and in microgravity conditions. So let me thank all my colleagues who worked with me to the development of these models, and thank you for the attention.
Speakers, if you'd like to make your way to the front, I'm sure people have got questions. I sure do. Thanks
Thank you.
very much, Monica. That's great. And actually, ESA has some questions, not just me. And what we would love to know, actually, maybe Marco, you can take this one, is how reliable and representative of human organs and tissues is the current state of the art in these 3D models that we're using?
It's a good question. It is very specific use. It's very specific to the term they use, the destination use. So there is no way to represent a full body or a full organ either. But for specific context of use, the selection of specific ingredients and the recipe for getting the tissues mature are definitely very representative. So the answer to therapies, to drugs, are as representative as animal models in specific circumstances, and definitely more than bidimensional cultures, or of course monocultures. The way to interrogate the systems needs to be as well developed, because there is no easy out-of-the-box way to analyze the systems, because are specifically realized for a specific use. So everything must be still defined properly.
So INSA using these, you're using these cerebral organoids. So how are these representative, or how far away do you think we are of those being a fully validated 3D model?
I mean, it's the best that we have at the present. So it's my very short answer. We could do better, maybe, and I hope we will do better in the future. But right now, that's as good as we can get, and it's pretty good actually, because we see that a lot of those actions that we see in the animal, we also see in these 3D models.
Yep.
So, yes, I think it's a pretty good system.
Mm-hmm, mm-hmm. And I guess the standardizing and standardizations around the world to create these systems is now helping a lot, so that we're all doing this.
Yeah,
doing
absolutely.
It the right way.
I mean, it's a big community now working on those organoids, and that helps a lot.
Yeah, yeah. So you were showing with your experiments a lot of like protein changes and gene expression changes. If you were to use these systems in a place where you cannot access them readily, you can't return the samples, what technology is already available for the in situ diagnostics that we might be able to use on those 3D models? And what's severely in need of development? Like, what do you want to see to be able to analyze your samples in deep space? And that's open to anyone.
Well, I'll start because I work with probably the smallest tissues. Definitely imaging. Imaging is still a very good tool to, and imaging is developing every year. So there are tools to also detect specific variations of cell behaviors. So that's the primary tool, but also proteomics. So you don't need to retrieve the sample, you just need to collect the supernatants, which is always accessible. So that is another possible tool to observation.
Yeah. Okay. So it sounds like so these 3D models are quite useful, and therefore that we should invest more in them. I'm taking that, of course. So we've seen organoids develop and spontaneously create these organoids. We've seen these tissue chips, organs on a chip. But what we haven't spoken today about is bioprinting. Now, Monica had a bioprinter with the leech looking at the wound healing, but do you see 3D bioprinting as feeding into the research and taking over? Like, do you think that's where the next step is, or do you still see value in these organoid and other models?
I don't think that the bioprinting will take over, because the point is you force cells together that usually may not have developed in the same environment, and I think that's pretty tough. But nonetheless, saying this, bioprinting is a valuable tool to also create the environment that maybe the organoids will thrive better in. So definitely, we cannot afford to say this is better or that is better. We have to use it all.
Yep.
So I would add that, so I don't think the problem is the bioprinting per se. I think the problem is the maturation of the construct you create. So bioprinting is a very nice technique that, in my opinion, allow us to create a lot of interesting models. The problem is that when you have printed something, you have nothing. So, because the problem is to create the conditions so that the cells inside the construct are able to mature, and to resorb the ink and create their own environment, their own extracellular matrix, and so on. And the reason, so for example, I like this model of the leech, and I like bioprinting in situ, because the graft is in an environment that create the conditions for so that the cell can work. Because this is, in my opinion, this is the part we should study and we should improve. How, for example, to give the mechanical and biochemical stimulations the cell need inside the construct. This is not easy, absolutely. But this is one of the problems I have to solve.
Yeah. So in situ applications, of course, there was the ESA-HRE project for printing on the skin, to print a skin graft, basically. So is your opinion then that it would be better to invest efforts into that sort of system rather than a 3D bioprinting, like that enables other research?
I don't think it's better to invest in one or the other, but we have to. So the gaps in knowledge we have in each system have to be clear to us because it's in that direction that we have to improve and to study.
Anyone else wants to chime in?
Yeah, just one final point. So one of the challenges, I think, with bioprinting is resolution. So it's very hard to get single-cell resolution. In fact, it's almost impossible to get single-cell resolution. And if you try and do that, thank you, you end up losing throughput. So you can't have high resolution and high throughput of cells at the same time. So it really limits the size of the construct you can create. The other thing is that I think cells are very good at self-assembling. So if you get the right conditions for cells to self-assemble into organoids, you end up with a better result because that's just recapitulating what natural biology does. I think there is definitely space for doing both, and you have to be careful about what scale you're thinking of talking about. But yeah, all models are models. There's no perfect model. We learn different things from different models.
Ladies and gentlemen, do you have anything to add to this discussion? Any questions for the presentations that you've heard today? Oh, we do. And I will go with the microphone. We just have the one spare. Thanks.
Thank you. So just because ESA gave already started with the difficult questions, I would like to ask how it is possible, for instance, with these organoids to study the main problem in space. So we have radiation, and this gives tiny effects. So if we have something like 2,000 organoids with I don't know how many cells, how it is possible to find some effect due to radiation, how it is possible to help to solve the problem of the astronauts' health.
Well, Mike,
Thank you.
okay. So my experiment in particular wasn't looking at radiation, but if you wanted to, then the thing is that you have to consider your organoid. Organoids generally go through quite a bit of medium. If you want to investigate that in space, you have to make a system that can sustain an organoid for however long you think it would take radiation to affect the bone marrow organoids, which, again, I don't study, but I assume based on the previous session, it was they were looking at 2D mesenchymal cells for like 96 hours, for 72 hours. So we see that already that cells respond really quickly to certain things. So yeah, it depends on your model system, but I would say you have to consider your radiation dose. Where it goes on the space station is also an important thing. But yeah, I'm not too much of an expert in that.
And that's the beauty of organoids. I mean, for instance, the cerebral organoids you can keep for years. So you can, if people would let me, I would keep the cerebral organoids for half a year or any length of the mission that you want to. So yeah, that's I think the beauty. And you have the statistical safety because you simply have the numbers. You can generate numbers, and that's I think what is the beauty about it.
Anyone else? Sure.
I've got a question for Ryan. So you showed a slide with your no-tox histofixative and kind of showing that it didn't work very well. I'm from a microbiology background, and I've found very similar things. I assume that you guys were kind of forced to use this no-tox histo due to NASA health and safety restrictions, as opposed to paraformaldehyde or glutaldehyde. But this morning, during the biofilms presentation from Katarina Sims at DLR, I saw that she used a combination of these aldehyde-based fixatives to preserve her biofilms. And I was wondering if you know, what would you change about your hardware, or could you change about your hardware that would allow you to use more appropriate fixatives for your samples?
Well, we can't change like the dimensions of the hardware in an exterior way, but if I had, and we were using like pre-made hardware from 20 years ago, but in a perfect world, then I would shrink it inside, send less organoids to get another layer of containment, and just get the paraformaldehyde, because I've tried 5, 6 different non-toxic alternatives to paraformaldehyde, and they all work varying levels of bad. No toxicity is least bad.
There was a restriction based on containment.
Exactly, yeah.
Yes, so first of all, my compliments on your work and your presentations. Really, really great work. As an ESA flight surgeon, it's very relevant for us to get to see these results. For example, the work on how the skin wound healing is affected by spaceflight. It has a lot of clinical consequences because we cannot just apply Earth-based medicine to our astronauts. We cannot use the same, let's say, protocols that we use on Earth, right? So I think that's really wonderful to see, and I think this has a future. We need to do more of this, and we need to do it fast because we still have a lot of unanswered questions. And yeah, we should send your brain organoids and basically all human organoids to deep space and let them sit at the gateway, preferably, for some time so that we finally can answer some real questions that we have on radiation and deep space radiation effects on a tissue that are hard to replicate here. So I really encourage you to continue this work, and also, as I heard, drug-related pharmaceutical effects, because we think that is also different in space, right? So, yeah, please continue work. It's actually not a question, it's more a comment.
Awesome. Yeah. Anyone can comment? Oh, we have one more question.
Thank you very much. Very interesting presentations. I just want to comment that not all cells can self-assemble, and we cannot have all organoids from self-assembling. Of course, it means that we also have to put some forces for 3D printing, bioprinting, and I totally agree that we cannot wait before we will have perfect, predictable concentration of cells in bioprinting when you already have so beautiful, reproducible results with self-assembling organoids. Of course, it's great, you can make it. About chip, I just want to ask you, taking in consideration that in the human gut we have 500 millions of neurons, and are you considering to add these cells to your chip?
Well, yeah, that's perfectly true. The gut, like most of organs, are very complex. Here we oversimplified, of course, because we wanted to target a specific application. We went on, actually, I didn't show the results, but we went from the gut organoid to dissect the organoids and see the cells inside the device, and that was already an increase in complexity because we actually could see a more heterogeneous population of different cells, more mature, more relevant. But of course, if you want to investigate the behavior of the gut based on neuronal effects or communication, you have to add another compartment which can emulate that. And that is increase of complexity, which needs to be motivated by the specific context of use, because that is to me, is the key word to build a new biological model which has a high complexity. So the complexity must be justified because of the specific reason.
And I would do absolutely the same with you. Just add cells. For example, is it, just wanted to check, is it human or mouse?
Human.
It's human, so it's a little bit more difficult. Mm-hmm.
So I would like also to say that the comparison between organoids and 3D printed products. So I don't know if this comparison is useful, because I think organoids are a very potent tool to study several phenomena and processes and so on. I think that bioprinting was conceived also as a tool to to be transported in some ways in the future in vivo to replace damaged tissues. So the concepts are completely different. But we have to consider that also from this point of view, we are very far, because also the 3D printed constructs now are very small structures in comparison to the dimensions we would need to apply them. So, for example, the in situ bioprinting in the leech was in this. So from this point of view, to apply in vivo to close a big wound with loss of substance. And we have results because the healing is faster. Obviously, we close the wound, so we close a door to the infection, you have less inflammation, and so on, and so you improve the wound. But, so there is a difference in between the dimensions of the leech and the dimensions of a human organism. So we have to work a lot to reach this objective.
Thank you. It is, I think, beyond 3:30. With that in mind, I'm going to wrap up this session. I want to thank our speakers, our panelists today for their inputs, for their research. I want to thank you, of course, for staying and listening to this talk and contributing as well. And with that, this closes session 3 of day 3. All that awaits is the wrap-up downstairs. So please enjoy the rest of the afternoon. Thank you.
So I